All right. Thank you everyone for joining. Today's webinar would be on career pathways in cryobiology, and it's hosted by Society for Cryobiology in combination with ICYR. So what is ICYR? Hi, I'm Lakshya. So I'm the current president of ICYR. And here is my colleague, Charles Elder, and he's also the vice president currently. ICYR is basically International Cryobiology Young Researchers. You can think it like a early career body for the society. And our main goal is to be the go-to place for any of cryobiology where the early career development is to happen. And we're talking about professional skill development. We are doing sort of multiple events, 4 in a year. For that, we have career opportunities, and will be a job career fair towards the end of this year. And then it's a place for social networking and building connections. And all of that sort of combines. Do check out the website, and we have all the events listed there. Apart from our sort of role outside cryo, we also host events at Cryobiology's annual meeting. And we have 3 events mainly coming up. If you're coming to the Cryobiology conference in DC, first event would be on Monday, 22nd of July. That's a welcome ICYR event. So come hang out, meet whether you're a lab technician or an undergraduate or a graduate student, a postdoc, or a young PI. We welcome all. So it would be good to see you all and just have that connection. Then we'll have a really fantastic sort of social tour on July 25th. That would be the ICYR July 24th on 7 to 10 p.m. And that would be the DC bus tour. The bus will leave right outside the hotel. So it would be a fun time and fun night to remember. And then lastly, we will have for our virtual attendees, if you're joining the CryoS virtual platform, we'll have a social event at the same time going at, on the same date, going at 11 p.m. Central Time, catering to European and Asian time zone attendees. So do check us out. And with that, also check out our website, and you will find, you may find interesting things which you may think as an early career cryobiologist. And we have Discord server and creating this community online where we can connect, moving from just once a year meeting to an sort of yearly throughout connection. I'll let Charles take over and introduce our panelists. Thanks again for joining.
Hi, I'm Charles Elder. I'm a PhD candidate at the University of Louisville and the vice president of ICYR. I wanted to start us off by introducing our panelists. We have Dr. Willem Valkers, professor at the Lower Saxony Center for Biomedical Engineering and the University of Veterinary Medicine, Hanover. We have Dr. Rebecca Sandlin, who's an assistant professor at Harvard Medical School and Massachusetts General Hospital. We have Kanav Khosla, Dr. Kanav Khosla, a scientist in drug product and process development at Sana Biotechnologies, and Dr. Pierre Comizzoli, senior program officer for research and veterinary research at the Smithsonian Institution. To get us started, we wanted to kind of emphasize that we're trying to make ICYR a hub for young researchers to find careers into cryobiology. So we decided to do this webinar where young cryobiologists can learn from some more experienced researchers. So to start us off, Dr. Valkers, could you describe your job and the duties in which you in which you're doing in your career today?
Yeah. Yes, just to introduce myself. So my name is Wim Wolker. So I did my bachelor and master studies and also PhD studies in the Netherlands. I studied molecular sciences with majors in biochemistry and biophysics, and then did a PhD in plant sciences, actually in desiccation tolerance of plant seeds and pollen. And after that, I did postdoctoral work at the University of California in Davis, the Center for Biostabilization. I had some various other positions that I will not go into, but currently I'm actually working at the University of Veterinary Medicine in Hanover, where I'm actually leading a biostabilization group in a biomedical engineering institute, which is focused on implant research. So this is a large institute with many groups from many different disciplines that come from either the medical school in Hanover as well as chemistry and engineering groups in Hanover. And I'm heading a biostabilization group within this institution. Yeah, what am I doing? My duties are acquisition of funding for my research projects, project management, supervising bachelor, master, and PhD students from start to finish, hopefully. I have some teaching duties as well, giving lectures in a master labor. Most of the focus from my research is actually on research rather than teaching. Yeah, I think that covers it for now.
Thank you. Thank you for giving us a brief introduction. And Dr. Sandlin, would you mind giving us some insight into your daily life and career in cryobiology?
Yeah, so I can start with telling you a little bit about my background and training. So I did my undergraduate degrees in chemistry and mathematics. And then for graduate school, I focused only on chemistry. So I worked specifically in bioinorganic chemistry, working in high throughput screening, design and implementation, and anti-malarial drug discovery. So nothing whatsoever to do with cryobiology. Then for my postdoctoral work, I was really interested in getting into something a little more translational. And I looked to biomedical engineering departments, and I found one center that was willing to take me, even though I had no prior training in engineering. And that was the Center for Engineering and Medicine and Surgery through Harvard Medical School and Mass General Hospital. And so the world of cryobiology, like I said, was completely new to me, but I found it to be really interesting. I loved the translational nature of the research, and I love that there's so much opportunity to really grow within the field. So after starting in the area focusing on ambient storage, I started doing more work in cryo. And eventually I decided to stay in that position, but as a faculty member. So I was promoted to assistant professor in 2021. And so now my research program really merges that prior training in chemistry and global health and malaria with more current topics in cryobiology. For example, my lab does a lot of work with cryptosporidium parasites and malaria parasites in the context of cryopreservation. So my daily activities are, pretty similar to any assistant professor, with one exception: I don't teach. I focus only on my research, and that's typical for all of the faculty members in my department, which is interesting. But I love spending time in the lab and working with trainees, and so that's where I focus my time. So managing the lab, mentoring trainees, either research technicians or postdoctoral fellows, and lots of grant writing and paper writing.
Thank you. Thank you. Dr. Camizoli, would you mind introducing yourself and talking a little bit about your career and your daily life as a cryobiologist?
Yeah, thank you so much again for the opportunity to talk to the next generation of cryobiologists. So I started my career I trained as a veterinarian in France, and then I worked for several years in Africa, and then I did a master and a PhD in life science. And at that time, I started to be exposed to cryobiology because part of my project was to develop freezing techniques for sperm and embryos of wildlife species. Mainly ungulate species. And then I moved to the United States to do a postdoc at the Smithsonian, where I'm still working. And at that time, I had to work on a new project of cryobiology in oocytes, using the domestic cat as a model for wild phthalates. And then I continued for many years to work on this projects of oocyte cryopreservation, but doing also a lot of services for the zoo, for my own zoo at the Smithsonian, but also for other zoos in North America to help to freeze really valuable materials from different species. And so now I still conduct research, not personally, because I don't really go to the lab as often as I would like, but I supervise students, postdocs, and scientists, and everybody is really working on cryobiology as as an umbrella, and we use cryobiology for multiple applications. But really, the main objective is to try to preserve biomaterials for the long term, of course, of different endangered species. And part of that now, as it's been expanded, it's beyond my lab because the whole Smithsonian Institution is interested in this kind of a cryopreservation technique. So we do have very large frozen collections, biological collections that I manage. So we have more than 1 million samples from 18,000 different species. So this is more like a work as an administrator of a large biobank. But it always goes back to cryobiology regardless, which is something that I like. And lastly, I still do a lot of professional training. So I talked about students we have in the lab, but we've been also organizing a lot of training courses on cryobiology in different countries. And that's been also very satisfying because it's been a way also to translate what we do in the lab and to diffuse our knowledge.
Thank you, Dr. Camazzoli. I've always been fascinated by the Smithsonian Institution, so thanks for giving some insight into your daily life. Dr. Kolsa, would you mind introducing yourself and talking about your career and your daily life as a researcher?
Yeah, thank you, Charles and Lakshay, for inviting me, and hello to all panelists and attendees. I'm joining from San Francisco, so it's still a little bit morning, so good morning, everyone. A little bit about me. I currently work at Sana Biotechnology. It's a biotech company that develops hypoimmune cellular medicines. So our final drug that goes into our patients are actually cells, living, breathing cells. And so as everybody here has experience in cryobiology, anything that requires cells that are off the shelf will require some kind of cryopreservation technique. And that's pretty much what my team and I do. We develop a process to harvest the cells, formulate them, package them, cryopreserve them, ship them. And in such a regulated environment, such as biotech and medicine, we need to develop, processes that are very robust. We need to do a lot of quality control. We need to manage any deviations that occur. We have to file lots of regulatory filings, perform studies to gather data to show that, our drug product doesn't change with whatever types of stresses that we put them into, and it's safe to be put into patients. And so Sana develops something very unique, which are basically hypoimmune cells. So these cells are essentially taken from a healthy donor and then gene edited and expanded in order to be put into patients and overcome allogeneic rejection, and then either have regenerative medicine applications or specifically for CAR T therapies, which is to fight cancer, mostly blood cancers, leukemia and lymphomas. And so cryopreservation is actually very important, a critical step in any of our process. Without cryopreservation, we can put in millions of dollars of research and capabilities into developing drugs, but we can't get them to our patient. And again, because manufacturing these drugs is such resource intensive, cryobiology really allows us to bring down costs of them by allowing one major run to develop, drug product for, let's say, 100 different patients. Currently, the standard of care has been to use autologous therapies, which is basically take immune cells from the patient and then transplant it back to them. That can cost anywhere up to $1 million, and hopefully allogeneic therapies, if they can overcome immune rejection, can allow us to reduce the product cost by, let's say, 10x. And so clearly, cryobiology, as I said, is very important, and that's what our group basically does. We have multiple drugs in our pipeline, and so my day is essentially It's a very collaborative type of place because not everybody is an expert in cryo. Some people are, process development, CAR-T folks. Some folks are from regulatory agencies. Some people are quality control. So it's a very collaborative, cross-functional type of space. So much of my work is obviously process development, but also communicating what that process does and how that changes our drug product. Some of the time we are actually manufacturing the drug product in, very clean room type of space. A lot of it is tech transfer because sometimes we have to transfer our technology to an external or internal manufacturing site. And as I said, product control, regulatory filings, that's also a big deal. So that's where most of my time goes. And a little bit about me, my background is in mechanical engineering, so I'm an interesting fit in biotech. I got my degrees at the University of Minnesota, which is also sort of a interesting place because it's a big hub for different types of cryobiological research that's happening. And that's where I used to work on developing cryopreservation processes, technologies, techniques for both model and non-model systems. So I got started with zebrafish, but then I worked on shrimp cryopreservation. I also worked on coral cryopreservation. So that was an interesting foray for me to get into biological applications of my research. And that's where I really got interested in cryobiology. It was also an interesting time when I was working there. I was like, some interesting developments occurred in the field that brought a lot of interest and capital to this area. And I'm hoping that that continues to go on with different centers and different young researchers now starting to, take up the mantle and move the field forward.
Thank you, Dr. Koso, for introducing yourself and talking about your daily life. Before we continue, I just wanted to, kind of give a shout out to ICYR again. We've put our webpage into the chat and some of the events we'll be hosting at Cryo 2024. I wanted to hand over moderation to Dr. Zhonghu Han, who will be moderating the rest of the event. Thank you so much again for introducing yourselves, and I think we have some really great questions to continue.
Thanks, Charles. Yes, for all the participants, attendees, please raise up your hand or directly put your questions into the chat. To ask your questions, you can ask your questions to a general, to multiple panelists or like a specific panelist you want to ask. Yes, Nick.
Yeah, thank you very much. So terrific to have you all on, and I know at least a couple of you pretty well. So this is a general early career question. Whether you're in academia or industry, there's this chicken and egg problem, right? Where you need grant money, but you need money to create data to get the grant and so on. So can anyone comment on what you've had success with or failure with when it comes to getting the resources you need to show the preliminary results that let you actually go for the funding to get the work done?
Okay,
Yeah,
so
there...
I can tell you, Nick, that sometimes it's a matter of being lucky. I say that because I got lucky, because when I joined my lab as an early career cryobiologist, there was enough fund already available for me to get started at least and to generate those preliminary data. But you're totally right. I think this is, but it shouldn't be, to me, I think it shouldn't be the responsibility of the early career scientists really to find, the startup money to generate those preliminary data. You need to find a good mentor.
Well, I am lucky in that. I've had some excellent mentors. Thank you, Pierre.
Yeah, I'm just going to add something that Pierre mentioned. And hi, Nick. Nice to have you join us. I will just say that, luck is very important. And I'm just going to be speaking mostly about my experience doing research and working in the United States. I know that's a very global society, but my experiences are mostly in the United States, so I can only speak to those. If you're doing good work, I honestly don't see that there is going to be an issue with funding. There is both private, government, and non-governmental funding options available, and I've seen many of our mentors being able to do that. I think the problem sometimes really just comes down to, finding the right problem to work on. Also, you have to be at the right place at the right time and the right resources, the right, collaborators, groups, in order to, actualize that kind of funding to happen. So I think you have to really match, the funders' interests into your research. And I think more and more translational work is probably going to get funded over the next few years through, at least from the NIH perspective and even from like the industry's perspective. So, if you're really interested in basic research, it's probably going to be a little bit harder, but I'm sure there is also resources out for that as well. But luck does play a role. And what we can really control is just making sure that we're working on interesting problems and problems of broad interest to the community, and we're really trying to solve a problem.
Yes, maybe I can add a few things. So yeah, luck is one thing, but you need a solid background, solid paper background, track record. That is of course important. That helps, but that's not the only thing. Even if you have a good track record, that does not always lead to successful funding. But you have to be open-minded to the topic. Don't fix yourself, I want to work on this or that. Just be open-minded. What is trending, so to speak? So you have to look on collaborating groups that are interested in a particular topic, and then you may have to adapt your choice. Don't fix yourself on a specific topic, but that is probably important. And this will increase your chances of getting funded.
Yeah, if I can add one more thing. So Nick, there are specific grants that are meant to be more exploratory, so you need less preliminary data. One example would be, once you're a PI, you can submit an ICYR 21. It's less money, but it's meant to be an exploratory project where you're trying to establish feasibility of the work you want to do. And the idea is, once you get the R21, you successfully complete it, then you'll have the preliminary data that you need to go forward and submit a larger grant like an ICYR 01 if you're in the US, for example. I think the other thing, though, is you can often repurpose data that you've collected for other applications. So, for example, I wanted to submit a grant for Drosophila cryopreservation, and I'd never worked with Drosophila in my life. So the first thing to do was to build a team. So find someone who is an expert in Drosophila, bring that person in, and then I was able to take data from my work with cryopreserving Anopheles to demonstrate that we have the necessary expertise to make this happen, even though we don't have any actual data for Drosophila cryopreservation. And it was successful. So I think taking that approach of building the team to show that you have competence to solve the problem, but then also trying to repurpose your data whenever possible to show them that you have the relevant prior experience.
All right, that's really terrific. Thank you, everyone.
Yes, and we just got a very interesting specific question. So from Nadia, so they have had experience with freezing human sperm, eggs, and embryos, and that they want to get some experience in ovarian tissue preparation. Anybody has any thoughts about that? I believe this is a very research-related question. But I believe we can still answer this question if anybody has any suggestions.
I'm sorry, I don't. Oh, sorry, Rebecca, you were gonna talk. Sorry.
Oh.
Okay.
Sorry.
No, yeah. Well, yes, it's a research question. I think it really depends where you are located on the planet, Nadia. But we can definitely, I mean, if you are looking for a place to get training in ovarian tissue cryopreservation, there are definitely a lot of different teams now doing that in human, but also in a lot of animal models. And again, by the way, that's exactly what Willem was talking about. Is you don't need to really you need to be open-minded, and if you're really your objective at some point is to do human ovarian cryopreservation, it's also very valuable to have an experience in animal models. So anyway, wherever you are, I think we can definitely recommend you a place to go or some people to connect with to get this training in ovarian tissue cryopreservation.
Yes, you can, of course, you can contact me afterwards if you have any specific research questions, but there's a variety of protocols available for ovarian tissue cryopreservation that go more in the direction of vitrification, and so maybe these details you can ask me afterwards. I don't have personal experience with ovarian tissue, but I have a good protocol chapter book that may be of help to get started in this field.
Sounds good. Thank you all for your answers. And here we have another question from Haiti. And the question is, how do you navigate finding postdoc opportunities, especially when you may not feel that your skill set doesn't necessarily match the work being done and you are willing to learn, but you are willing to learn? Yes.
So I can give some advice. Don't hesitate to reach out to individual PIs. We may not always have funding to support a particular postdoc, but anytime I get a good application in my email box, I'll spread it around to other faculty members who might be interested. And I would say most of the people who come into our center, that is how they've been hired, is they just sent an email to one of the PIs. And even if that PI couldn't hire them, maybe someone else in the department could. And depending on the type of department or center you're applying to, having a different skill set is not a bad thing. So we often take people from other backgrounds. So we have biologists, molecular biologists, chemists, engineers, including mechanical, electrical, biomedical. And really that makes the work a lot more exciting. So we're specifically looking for people with different backgrounds. Now you have to have like a common skill set that overlaps with ours, whether that be just research in cryobiology or some other niche application. But don't hesitate to reach out to people, even if your research doesn't overlap with theirs.
I will just add to what Rebecca said. I will just say you've got your PhD. Have faith in yourself. If you can get your PhD in one area in one specific niche, you can spend another few months to another year or so to learning a different field, at least getting the basic competencies. So again, don't hesitate to reach out to different people. Most of the PIs are willing to mentor students. Some labs specifically geared to hire different types of people with different types of skill sets. So there might be other postdocs and other students, technicians that might be willing to help you. So again, you will miss all the shots you don't take. So it's worth taking that shot.
Yeah, I can echo on that. So what is important, write it a little bit personal. So show that you actually have studied what, for example, if you're applying with me, that you know what I'm doing or have at least some idea. Because I get a lot of very general application, and usually I don't respond to them, to be honest. So write it in a personal way. And even if I don't have funding, that increases the chances that at least we can discuss and think about applying for a fellowship or things like that. There are always opportunities also for foreign countries, special collaborations between Europe and other countries that there's funding opportunities. So try to write it personal. And yes, even if I have a topic that is related to cryobiology, I would rather look at people that have different skills that complement what I'm doing. So that can be some kind of programming, that can be engineering or anything. I don't look at people that can freeze cells with 10% DMSO, but I look at other things, other skills that complement what we are doing. So that's how I look at it.
Thank you. And I believe Haiti has a follow-up question. Laksha, do you want to unmute them and they can just ask? And I think they just raised their hand. Yeah. Are you able to do that? Yes, there you go.
Yep. So yeah, thank you all for that response. I just really wanted to ask that question because I'm getting ready to defend come December, but my focus has really been in optical microscopy and measuring things as they enter plant tissue. So when I see cell and tissue culture for most of the million folks who are out there posting postdocs, I get a little bit nervous about applying to those positions. So I really do appreciate all your feedback.
Okay. Yes. We got a another question from Nusa. Where do you guys usually go to find funding opportunities? And well, they are interested in preservation of the plant biodiversity. And yes.
I think it depends on where you are. So I'm in the US. So when I go to look for funding, I go to usually governmental agencies. So the National Institutes of Health, the National Science Foundation. But then I also look to make connections in other ways. So for example, I also work with the Centers for Disease Control and some industry collaborations. So in those cases, they don't have specific grant opportunities that are posted for the public to apply to. But if you meet people there that you can collaborate with, there are opportunities to find internal funding through their organizations. But if you're just looking for new funding opportunities, in the US at least, you typically would want to go to governmental agencies like NIH or NSF.
I'm just going to add a little bit industry startup type of perspective. The NIH is mostly for human medicine, so they're probably interested in those types of associations. So, probably USDA, again, USDA doesn't have the kind of resources that the NIH has, the budget, I mean. So obviously that could be something. But again, there's very interesting work being happening in the private sector and in the startup field. Obviously, many startups do fail, but you end up learning a lot from that type of experience. So I'm just do quick Google search, right? There are tons of companies right now working on plant genetics, whether to make them more resistant to pests, whether you're trying to improve yields. I just looked up a company called Ohalo, which is in the Bay Area over here, and they're hiring. They may not hire for a cryobiologist, but you should just keep tracking those types of opportunities. They may want to collaborate with your lab. They may want to be interested in your type of skill set. It's very cheap for them to offload this type of stuff for $50,000, $100,000 type of startup grant. But again, if you have the expertise and you have the skill set, you may be able to find something if you keep looking up. So think of non-traditional ways of actually doing this type of work as well.
Thank you for those great answers. And we have a more general question from one of our organizers, Nicole. Could all the panelists speak about what attracted them to their current sectors and what are the pros and cons in their chosen sectors? For example, benefits like retirement, career progression, etc. And is there anything their sector has or does not have compared to others? Yes, anybody want to go first? Dr. Komisarly?
Yeah. Well, yeah, but okay. So my case is very very specific, but so I work for the U.S. government, so that means that part of my activities are supported by federal budget that is allocated to the Smithsonian every year. I would say that the money that we receive is not enough to do all the research that we need for cryobiology, but I was referring to the management of the frozen collections and the maintenance, and at least that covers the basis. I would say that that's the pros of working for a large agency or organization is that at least you get what you need in terms of the basic needs, Then after that, the cons is that working for a large organization, then you face a lot of administrative hurdles, and compared to I'm looking forward to hearing from my colleagues who are working in the private sectors. But for us, sometimes it's very, very slow, to get something new started or to buy something, and so that's a little bit frustrating. So that's my own experience.
Can tell a few things about Germany's situation. In Germany, I think generally the benefits, our health, social security system is actually really good. Salaries are more or less regulated if you work for a government or state organization. Yeah, but industry, in an industrial position, there's more variation, and you can maybe get a better or higher salary. But generally benefits are really good, I say. But yeah, of course, the most important thing is to secure a permanent position, and yeah, as that is probably one issue that is. And if once you have that, then I think the situation here in Germany is quite good.
And Connell?
Yeah, so I was thinking about this before I was coming into the panel, and I guess you really have to be honest about what you really want to do with your time, because you have limited time, and you gotta really do a self audit in terms of what energizes you, what motivates you. I think that would be my biggest advice to all young researchers here, young early career scientists here. And so when I sort of was doing that in early 2020, 21, obviously with the pandemic, those things are on your mind. I started to figure out, what was something that really energized me. And I think that's been true for me for many, many years. It's always working on major, big problems, important challenges. And that's why I was interested in developing solutions to protect the Earth's biodiversity. The impact of my work is just, something that's important to me. That's why I pretty much got my PhD, stuck along for quite a bit in academia. Is really, I want to have, my work should have a high impact, and it brings a lot of meaning to me. So obviously it was going to be a place where I was going to be working in new technology development. I just obviously knew that. But then I also realized that I also really enjoyed working with people. I was an outgoing type of person. I liked, working on cool types of ideas, different types of ideas, not necessarily in cryo, but like all different types of applications. And I just noticed that when you're an early scientist, you're a postdoc, graduate student, most of the work is happening either by yourself or maybe with a very small team. So that really didn't work well with my style. And so I just noticed that, maybe those things were at a conflict for me. Obviously, academia allows you to have a high impact, but maybe doesn't allow the collaboration you may seek. Also, it does not provide the kind of economic security I would want for myself. And I also didn't want to spend any more time being a trainee. I wanted to be, have my own career and take ownership of it. So I just thought that was probably not going to be possible in academia for me. Also, academia is, if you just, we're all data-oriented people here. It's a numbers game, right? There's very few positions out there. Hundreds of people applying for grant applications. You will lose out on many of those applications. Whereas I'd rather, just go to a place where my skill sets were valued. I still had the opportunity to make a huge impact. Again, what I'm doing right now, we're trying to cure cancer, trying to come up with ways to overcome immune rejection. Those are big scientific problems with huge impact on society. And so my job allows me to do that. But at the same time, it allows me the collaboration I seek. It allows me the opportunity to have a more economically secure life than as an early scientist or a postdoc, for sure. Living in America, obviously I live in the Bay Area. You guys can only imagine if I was a postdoc here, how hard that would be. So those were really the three major things I think that pushed me to, kind of make a transition out of academia and move towards industry. And I'm very thankful that those opportunities were there. And I get the sense with changes in the in the bioeconomy, there's new types of biological companies being opened up with different types of biological products. You guys should just take a look around. There is cellular medicine is big, cellular agriculture is big, and all of those companies, those applications will need some type of biopreservation. So, your skill set would work really well there. It's just sometimes it's the right place, right time. And again, I would just tell people to really do a self-audit to try to really get down to the motivations behind what motivates you to be a cryobiologist or engineer, scientist, or researcher, and try to make it happen for yourself. Because again, you own your career, nobody else does. So you have to make those decisions.
Thank you, Connor. Becca, do you want to share your thoughts?
Sure. So this is a really easy question for me because I spent a lot of time thinking about it before deciding to stay in academia. The pros are I can do whatever research I want. I mean, there are some limits, but if I wake up tomorrow and there's a completely new organism or cell type I want to work with, there's no one that will stop me. I don't have to ask permission. I can just do it. The con is I have to pay for it. So that's what takes a little more work. But yeah, that's definitely what drove me into staying in academia and what I love to do. And I would say that I do a lot of collaborating. So I would say almost all of my grants have multiple PIs on it, and most of my papers have multiple PIs on them. So I actually collaborate a lot. But again, yeah, the pro is I can do what I want. The con is I have to pay for it.
Thank you. Very straightforward answer. Thank you. Thank you all for all your great answers. And I believe we have a anonymous attendee and who seemed to be a undergrad student. So they're asking, how do you know if you should pursue grad school? Yes. Any panelists want to answer that question?
Well, that's, I mean, if you really want to, here we've been talking a lot about research, development. I mean, it's obvious that you can't really do that with just an undergrad degree. You really have to go to grad school. You don't necessarily need to do a PhD, but at least to do a good master. And there are some professional masters that help you to then after that access to some good to have good opportunities to get jobs in the industry. Or but I think it's, to me, it's not I mean, yeah, that's the if you really want to do exciting stuff in cryobiology, you definitely need to go to grad school.
Yes, agreed. Any more questions from the attendees?
I would say if you haven't
yes, sorry.
if you
Go ahead.
haven't already, you should do some type of internship so you can actually do research in an academic lab. And then that's probably going to be the answer you need, because grad school is going to be very similar to that work that you're doing as an intern.
Yeah, I think that's a really good piece of advice. If you can, let's say you live in a big city, could be Minneapolis, it could be Boston, try to get into some of the labs, or try to do a summer internship, or maybe even for a year-long type of technician position if you could get, and really see if that works well for you. Because yeah, majority of the time in graduate school is spent in the lab, specifically for the kind of applied biological sciences type of work that cryobiologists do. So, and again, cryobiology hasn't translated that well right now into industry. So obviously the opportunities to be in industry will be quite few, and there are still labs out there. So do that. I would also just recommend that you probably want to move or be in a place or an ecosystem that is really going to help you, identify that. So, obviously university is a place where you can look at different, you have a lot of different opportunities. You can work in different labs in different areas in different departments, and maybe in a short period of time, you can find, things that may be of interest to you and build skills in that area. So it's a really, the university is a nice place to do some type of like soul searching, discovery, getting more and more insight about yourself.
Yeah, what I wanted to say is that depends a little bit also if you're in the USA or in Europe, if in terms of when you stop your graduate studies after your master or PhD studies. So the further you get in your graduate students, so suppose you choose to continue the PhD path, the more narrower your topic will be. So you must realize that once you go the PhD path, you must be open-minded to move to any position worldwide. because the more specialized you will be, and yeah, as long as you realize that, so then I would consider a PhD. But if you want to stay close to home, or yeah, you may want to stop after a more general, let's say a bachelor or master degree, and find a job in industry. So depends on what you want.
Thank you. Great answers. Yes. Yes. Our next question is from Mohammed. How partnerships with international organizations and collaborations can be developed in cryobiology in developing countries? Very specific. Yes.
Well, that's a tough question. Well, it depends again. It depends on which developing countries, but well, let me give you just an example that might not be really true for everywhere, but I've been working a lot in Southeast Asia. And because it's, of course, Southeast Asia, it's a hot spot in biodiversity. There is a huge need to cryopreserve a lot of biomaterials. And but there are countries like Laos, Cambodia, Myanmar, Vietnam that have a little bit of resources, but they don't really, it's really difficult for them to get started. But luckily, in that area, there is a country that's a little bit more advanced in terms of education, schooling, and money for research. It's Thailand. And so we've been using Thailand as a hub to train a lot of people and to get them started really with their cryo projects, and then after that translate and transfer the technology to their own lab. Now you have other parts of the world like Africa, where it's going to be probably much more challenging, because in Africa there is, of course, there is cryobiology in probably in human reproductive medicine or in human biology, but there is not a lot of strong research lab because of the lack of funding. I'm not going to talk about all the whole planet, but the take-home message is that you need really to find really local, regional partners from countries that have the possibility to train you and to get you started, basically.
In Germany, a lot of these collaborations go via the DFT, so that's the German Research Foundation, and you would have to look for calls. And I know, particularly for developing countries, I've recently seen various calls where there's a collaboration between a German institution and another country, and you can apply for funds. So these open up some opportunities, so via national funding agencies.
Thank you. Yes. We have a next question from Harry. As a current PhD student with cryopreservation experiences and interested in pursuing a industry or government career in cryobiology, are there any advices for reaching out and searching for such job positions? I believe, yeah, Kanav, you want to start?
Yeah, so again, I can only talk about the US, and in America, all of this is gated by what your visa statuses are. So that you have to, not everybody is like as fortunate as I am to, be able to, overcome those types of things. But there's also, so any kind of government job will require some kind of residency or US citizenship. So if you're an international student, which many of you guys seem to are, it'll probably be very hard for you to break into that field. But again, certain opportunities might be there, you might be good, so they really can't ignore you, and they will make changes in order to make that happen. Same in industry as well. I think the market is kind of going through a contraction, and so you'll see the number of posted positions will be going down significantly. But over the long term, I think this will come up, and maybe hopefully there'll be some changes in the US immigration system as well that may allow talented students who are here to be able to get those opportunities. I would just try to, expand your network as best as you can. Speak to those people. Let's say you're in graduate school right now and you are still maybe 2 or 3 years out of defending. Maybe you want to just keep tracking of what positions are out there, try to, save them in a Google Doc and look at, what the job descriptions really are looking for. And then maybe try to tailor 1 or 2 experiences that fulfill those types of, needs of that company. Again, people are looking to hire because they want to solve a particular problem or have a specific need for something. So if you can match that at the right time, then I think you are positioned yourself for success. So that's what I would recommend. If you're a US citizen or resident, things are a little bit easier for you. I know that there is a general fellowship in policy that you can apply for through the AAAS, and you can become a fellow and work in government policy for up to 2 years. I know all the different government agencies have introductory level positions, anywhere from DARPA to NIH. So that might be an easier route for you. But again, if you're looking for an industry role, you also have to make sure your skill set and experience are basically geared and set up for what you will be expected to do, at least in the beginning in that industry position. So the reason why I got this position is because I had over 8 years of experience in cryopreserving things, and they needed a cryobiologist to come in and develop processes for cryopreserving cellular medicine. So it just perfectly fit. But if you have a certain type of skill set that a company is looking for, then yeah, things will be easy. Otherwise, it's hard for you to make that type of change. Again, there are some big companies also, such as Intel and other semiconductor companies, that hire lots of PhDs because they have very complicated problems in manufacturing and robotics. And also, I think with AI, things might be a little bit different too. And if, again, you have more of that kind of skill set, it might work there, going there as a generalist PhD. But again, if you're a biomedical scientist, biotech is probably your best bet in industry, I would say. So just keep a lookout for, what people are looking for. Mostly I noticed that if you're good with your hands, you can culture cells. That's like everybody needs to have that skill set. So if you are in your lab, make sure you're doing those things as well.
Thank you. And I would like to add one more thing on that, on top of that. And I think the ICYR is creating a flyer about the job, how to find jobs. We have like a few useful links that can help you and guide you to find jobs. Yes, and our next question from Bibiana is again a very specific research related question. So they work with plant cryopreservations, and they use nanoparticles to preserve their samples. What about the use? They want to ask, what about the use of nanoparticles in human tissue cryopreservation? Any insight on that? If yes, that happened to be like one of the field works that our lab is working. If I can help answer that, yes, we are using like some iron oxide nanoparticles to cryopreserve human tissues and organs. Yes, and our due to the interest of time, we are probably gonna be limited to like 1 or 2 more questions. So next question is from Mark. What is the total compensation like a rough range for a recent PhD going into postdocs or as a cryobiologist scientist?
So I can comment a little on that from the Boston area perspective. So Boston is a huge city. It's very, very, very high cost of living. Going into a postdoctoral position right now, the rates are going to be around $70,000 per year. The universities have recently increased that number. When I started 11 years ago as a postdoc in Boston, it was $39,000. So it's come a long way, and that's very, very recent. And I don't know if all Boston schools are doing that. But the NIH also just passed new rules that the postdoc salaries will also be going up. So we're consistent with what the NIH is going to be doing. So yeah, $70,000 a year high cost of living, though, 70,000 doesn't go far. And if you're going into a starting entry-level industry position with a PhD, and let's say 1 year of postdoctoral experience, because I've had a few people do this, I think their starting salaries in Boston were usually between 90 and 130,000, but this can be all over the place. So the higher end of that would be, this person had a special set of skills that were needed for the position and they were just the perfect match. The lower end is probably more likely, but the more experience you have, the higher that number is going to be. And then I think Conov would probably have a better idea beyond that.
Yeah, I would say that number seems obviously going to be inflated for San Francisco, which I can talk about. And also our company has postings in, it's located in Seattle and Boston as well, which are also other major biotech hubs. And you're competing with talent with all these different biotech companies. The market right now is contracting, but let's say over the next 5 or so year run, I noticed that there will be an upward trend because again, cost of living is going up. So yeah, you can expect anywhere from $100,000 to $140,000 as your starting salary. Again, depends on your level of experience. And again, I think once you get into a company, you understand the way the things are working, you can, sort of find your niche, your area, and then grow with it.
Would be good. I don't have
Glassdoor.
anything to add because I'm in the US, but would be good to hear from Willem since it's in Europe.
Well, I can say probably a starting salary for a postdoc is around 60,000 euro. But you have a lot of benefits. So your health insurance, etc., is covered, etc. And the public transport is almost free in Germany. So that these are some benefits that has to be considered as well. And of course, your salary increases. It's fixed, by the way, in Germany. So wherever you are, and even though in some cities, Munich, the cost of living is much higher, it is, if you are in academia or a government position, the salary is fixed. But generally, some of the other benefits are quite good here. So, and in industry, there's much more variation. So it can be. It can go in either direction, but fixed through the country. So not in cities that are more expensive, you don't get more salary, which I believe is the case in the USA.
Yeah, just Glassdoor.com is a good resource if you want to look at different salary ranges for different positions. So you can look at entry-level scientists, engineers at different companies and get an idea. And it does change by location, so you can get all of that information there. Might be not as accurate, but you'll be within 10 to 20% of the actual number.
Thank you. And due to the interest of time, I would like to ask one last question to all the panelists. And that question would be, what is success to you? And what is your suggestions what advice you want to give to a young cryobiologist? Yes.
Oh, that's an interesting question. So that you sent out before. So it depends a little bit where you are in your career. For me, in the beginning, when I started, success was doing an exciting experiment in the lab and something that had an expected outcome, then you were happy, or something that had an unexpected outcome and that you had to think of a new model or a new idea or a new concept that contributed to your specific field of interest. So in that, their success is defined by your laboratory or can also be theoretical work, right? And later, success is more defined by papers, then by more papers, then by citation. Then you realize, oh, my papers also need to be cited, otherwise who cares about what I'm doing? So citation, H-index, and then eventually success is defined by getting funding for the things that you like to do. So, and for me also success is defined by supervising master projects and bringing them to completion from start to finish. So that also is a definition of success for what I'm doing.
Yeah, so briefly, I would say I totally agree with what Willem said, because I'm also in the research academic, more or less academic research. But success to me, it's being satisfied by your job. No matter what you have done and no matter what you do, is to be happy. And that's to me the most the most successful career that you can have, is to be happy with what you've done. And of course, to get some because you have, because you got some recognitions of your work, not only in publications, but the impact of your work. But and then in terms of giving advice, I think we all talked about that. Don't be very curious. Do not really don't be stubborn, and be open. And if you cannot really do something that you would really like to do immediately, well, it's not a problem. You can, as Rebecca said, it's good to have a diverse background. You can try something else and gain some more experience and in other areas, and then go back to the area that you like. So you will get stronger. And so don't give up, and be curious, and be happy.
I guess I will just add three things that I think sort of define success for me. One of them is obviously doing meaningful, impactful work, which you can get from your job if you're that type of person that drives a lot of meaning from their work. And I think I have that right now currently in my life, and also did when I was in graduate school. The second thing is obviously your mental and physical health. Graduate school and academia is very grueling, very competitive type of place. So I would really recommend everybody to have one or two types of things that you like doing. All universities have beautiful parks, gardens that you can walk in, that you have beautiful recreational centers, specifically at all the Big Ten schools, I know. So spend some time there, try to get in really good mental and physical shape, because that will help you both in your career and in life. Specifically in terms of longevity, life is really a marathon. So really focus on that, try to get good sleep, eat well. I know young people sometimes don't eat well, or don't sleep as much or drink too much. So I would actually refrain from that and really cherish this time where you're young and have the ability to work out and do fun things. So focus on that. And then third thing is, again, happiness really comes from being in the company of people you like and enjoy. So if you can find that core group in graduate school, outside of school, just try to spend as much time you can with them. I think those are really the benefits of success, right? You have earned your time to be with people you love and you enjoy being with.
Becca, you want to share your thoughts?
Sure. So first, success to me. So, there are markers that have been, or benchmarks that have been set by my employer on what success is. So trying to meet those benchmarks is obviously very important. So these are things like publications and grants and awards. But I think the success that brings me more satisfaction in my career is seeing my trainees move on to the next stage. So when my research technicians get into the grad school they wanted to go to, or when my postdocs are able to find either an academic or industry position that is meaningful to them, then that makes me feel like I've been successful in my job. As far as advice for cryobiologists, young cryobiologists, I would say make sure you're paying attention to the other science going on around you. There's lots of exciting work happening in other fields, and you may find inspiration from those that you can pull into your own research program. So cryobiology is great, but make sure you're paying attention to some of these other research breakthroughs that are happening.
We'd like to thank you all for coming. We're a little bit over on time, but this webinar was very informational, and we can't thank you enough for sharing your experiences. Thank everyone for attending. I wanted to give a shout out to ICYR. We shared the website link in the chat. If you'd like to join ICYR, there's a registration form on that website. If you're a PI or a business owner, please recommend your employees or your students to join ICYR. Again, we're trying to build a hub for young researchers to find careers. So thank everyone so much, and have a great day.
Thank you, everyone.
Yeah. Thank you again for joining.
Thank you all.
See you.
Thank you.
Bye.
See you.
See you. Bye.